Okay, Al, you ready to roll?
Yep,
Okay.
and rock.
Okay. So welcome everybody to the fifth and final talk that Al is presenting this evening. How far did you get, Al, in this list? Like about here?
Right there.
Yeah.
Yeah. I did exactly what I said I was
You
going to do.
made great progress.
Yeah. Oh dear.
I'm just teasing. Anyway, so thank you once again for coming out on such a lovely evening to listen to Al's talk about science. And I just want to thank all of you for supporting the library and for all of those who helped Al and Denise put this together, those who did all kinds of work in the background like Kim. And I think it's been a real privilege to learn from you. And I think hopefully we will be able to do this again, perhaps,
Well,
in the fall. See if you can get to the next couple of stages, eh, Al? Anyway, I will turn it over to you.
okay, thank you very much.
Claire?
We're not getting to the end of the world today?
Just the beginning,
What's this?
the beginning of the end. The beginning of the beginning.
As Churchill said, we may not be at the end of the beginning, but we are at the beginning of the end.
I'm gonna say something.
Well, okay, so clearly I have not quite, oh, nice to see you, Aaron. Hope you had a good time in Costa Rica. That's great, nice to see you back. Yeah, okay, so I admit I have not quite finished. Could we see that schematic on the zigzaggy one, the one that Kim came up with?
Oh, yeah, for
Oh,
sure.
can I see it then, please?
Sure,
I don't
hang
want to
on.
prevail unduly upon you, but,
No.
Oh, wow.
It's up, Al.
so you're not going to show it, okay.
Oh, it's there,
It's up,
it's
Al.
there. It's up.
Oh, okay, thanks. So we're here. We're not quite to there, okay? I want to show you an obvious mistake. That's a Dickinsonian. The reason I mention it is because I looked at my news feed yesterday, and it said, latest news, identity of mysterious animal finally cracked. Well, it's that. They've known since 2018 that that was an animal, but they're presenting me that news on my news feed. Latest thing, it's an animal. Oh, I knew that. Anyway, this thing says that's 800 and something million years old, and that's wrong. Should be down here at 550. But other than that, this is where we are. Tonight, okay, this is what I'm going to do. I got one last interesting thing to tell you about camels, unbelievably enough. Then I want to make sure you know the difference between a prokaryote and a eukaryote. All the other stuff I talked about, I will not talk about again. I underlined everything on the board that's really important in green. So, we know that life has produced more chemicals, more rocks on Earth than any other thing. There are going to be 6,000 rocks on Earth. Life made most of them. We know that if we're going to get into an ice age, it's tilt that's going to put us into the ice age, not the other two elements of the Milankovitch cycle. You now know the difference between mafic, metallic, and felsic, light rocks. Felsic rocks are granite, mafic rocks are basalt. You know the difference between igneous, sedimentary, metamorphic, and just to make sure we all know, organic. It doesn't mean natural or good. It means in chemistry, that type of chemistry in which organic molecules get together with other organic molecules, mainly carbon. That's it. It's about carbon. And about carbon as well, two important things about it. It controls the temperature of the Earth more than anything else. When there's a lot of carbon, it gets too hot. When there's not enough carbon, it gets too cold. The first two ice ages were caused by there not being enough carbon. The biggest extinction in Earth's history, right here, happened because there was too much carbon dioxide and the Earth got too hot. So it's either one or the other. All these extinctions have always involved that, carbon dioxide. There's lots of other factors, but anybody that tells you that carbon dioxide isn't the deciding factor is lying. What else? Eukaryotes and prokaryotes, we'll do them in a sec. Macromolecules, I'm just going to be talking about them from now on as if you know what I'm talking about. Proteins, lipids, carbs, and nucleic acids. I'll be dealing with proteins and nucleic acids tonight when we do DNA. Lastly, of all the, sorry Craig.
Okay, no, it's all good.
Here. There. Of all the molecules in nature, the most important are these. They're called macromolecules. And tonight, I'm gonna show you how proteins are made and a little bit about nucleic acids. So, that's it. Okay, camels. Poor Denise, she's got a terrible job because I have tried day after day to go through this lecture, and each time I do it differently. So she does not know what to expect. And honest to God, this has driven me nearly nuts, so I will need a break. Because, you know, when we were doing Alexander, I digress all over the place, okay? I admit it. I digressed all over the place. When we got to the Caspian Sea, I was more interested in the Caspian Sea and the size of the sturgeon in it than I was about what Alexander did when he got there. Yes, I admit it. I digressed. Shamelessly. I fall upon the ground. I repent. Okay? But with this, honest to God, I've hardly digressed at all. Because when you get into something like life, you go to talk about one thing, and it's so hard because other things keep coming into it. I go to tell you guys about something, and I think, oh, wait a minute, I better tell them about that first so they'll know what I'm talking about. You go to tell them about that, and I think, oh, they need to know about that. And it just becomes like Russian dolls, you know? So that has been driving me slightly mad. Well, I guess quite a bit, right? But tonight we're going to get through it all, and then if we do this again, which I hope to, then we'll take it period by period, okay? I think probably when we do the Archaean and the Protozoic, we can do them together, because it was called the boring billion, and there wasn't that much that happened, so I think I could fit all that one into lecture. I mean, there's still stuff, but I could fit a couple of periods. I could put probably 1 billion years of Earth history into one lesson there, if I did it properly. With the Ediacaran, though, and the Cambrian, you get down in here and things are really happening. You've got all this complex life in the Cambrian. Then in the Ordovician, you got this big extinction. Then the Silurian comes, another big extinction. Coal forests and the Carboniferous, that's where all the coal in the world comes from. Big stuff in the Permian, biggest extinction ever. Some of these things are going to take me a while to cover. I mean, seriously, if I were going to do it properly, it'd probably take me a couple of years. You know, because I don't know how else to do it. I thought I could kind of whip through it, but if I'm going to do it properly, this is the only way I know how to do it. So, yeah, okay. Camels. See these guys? Where'd they evolve?
North America.
Ha! Good man. I'm encouraged. Thank you. Okay. 44 million years ago, camels evolved in North America. Then some of them went by the Bering Land Bridge. Can we just have a map on? Any old one will do.
Any old one?
Yeah, a map.
I'll just give it a try.
Give it a, yeah, okay. So camels evolved here in North America, 44, then they went over by the land bridge and they came over to Asia. Where did they live in Asia? They lived right here. Why? Because that's a big desert. That's the world's highest, rockiest, and coldest desert. Desert was formed when 100 million years ago, Madagascar and India left down here. India went north, rammed into Asia, created the Himalayas 50 million years ago. That changed the whole weather system of the Earth and eventually was a contributing factor to our ice ages. It started about 30 million years ago. So changed the whole weather pattern of the entire Earth, sucked all this heat in off the ocean, up into the mountains, rained there, so it's wet here and it's dry there, and that's when the camels went. Okay, we all know that. Well, eventually, North and South America joined, and the camels that have been in North America all went extinct. But what did we have left in South America? Let's see that previous shot on the one with the camels.
With the camels?
Or 146 will do.
Well, you're gonna have to go
No,
one at a time.
Okay, what-
146. Okay.
But I need 146. Yeah, there it is. That's a llama. That is a camel.
They're so cute.
When the camels went to South America, they evolved into llamas, guanacos, and the other, you know, the 4 cameloids that are down there. Yeah, well, that's a llama. So if you wonder why llamas are so tough, that is because camels are one of the toughest beasts on the face of the earth. That's not one, but remember the camel with the 2 humps? The Bactrian camel? Those things, as I said, live in the Tibetan Plateau and the Gobi Desert, and there's a 100-degree temperature difference there between the summer and the winter, and those things take it. They are some of the toughest animals in the world. And the last, unanimously, but good to know I'm agreeing with. Right.
Just speaking the word llama terrifies the dog.
Yeah. Those llamas are tough, man. Like, we watch BBC Earth all the time. You watch those cougars trying to bring down those llamas. I mean, a lot of the time, the cougar gets the worst of it. Those things are tough. Anyway, here's the last thing you'll ever hear about camels. There was a gold rush in California, and camels were imported into southwest United States by the U.S. Army because it was thought they'd be a good thing to pack supplies around with. That didn't work out, so the U.S. Army sold the camels. The camels were bought by 3 British Columbia businessmen. And do you know what one of their names was? Heffley. Adam Heffley. Ever heard of Heffley Lake?
Yes.
Or Heffley Creek?
Yeah.
Those guys brought in a whole herd of camels from the United States, put them on the Caribou Wagon Road, whereupon the camels terrified the mules and the horses. And you actually had mules jumping off the Fraser Canyon, fully loaded, because they panicked on smelling and meeting the camels. So the camels didn't work out. So they were let go. One of them was shot and served up in one of the prominent BC hotels. But there were mysterious camel sightings in southwestern BC for 20 years after that. But that's not the end of, yes?
I have a camel question. The Bactrian camel has 2 humps.
Correct.
Does that mean it can store more water and fat? Is it tougher than the dromedary?
I don't know. But I do know that this is the camel that the breed breeds true to. Dromedaries have 1 hump, but they have 2 humpedness in them, and they will produce offsprings with 2 humps. The ancestral camel still contains the genes for 2 humps. People took Bactrian camels, and as far as I know, Diane, domesticated them into dromedaries, and they were eventually introduced into Africa about 1000 BC. They're not native there. We always think of them, you know, people in African camels, but they're not native there. They're brought by people. Anyway, the last thing I want to tell you is this, and now I'm going to get to say it finally. I've been trying for a month. After they didn't work out in BC or California, Australia had a gold rush. The biggest nugget ever found of gold weighs as much as a human being, and that was found in Australia about 1865. What did the Australians do? They brought in the camels too. Camels didn't work out there either, but the camels proliferated in the Australian outback. Just think about it. You've got the Australian outback, like one of the biggest deserty areas in the world with almost no people, and it is now a raging desert full of camels. The camels just love it. Now, Saudi Arabia, they still race camels in Saudi Arabia. Guess where they get some of their camels from?
No.
Australia is selling camels to the Arabs. Isn't that great? Okay, that's the last thing I wanted to tell you. The other thing is about prokaryotes versus eukaryotes, because we're on to science now. According to this one, I might need page 100 or number 100.
No, 77.
There it is.
She knows her stuff. Oh, wow, that's lovely. Okay, the difference again, now we're not going to go into a lot of detail here. What I've been trying to do in this, whatever you want to call it, is to try to introduce concepts and then try to come back on them. Like, that's a tried and trick too in teaching, right? You introduce something and you repeat it, and then you come back to it and you introduce it again, add another layer to it, and you build up that way. That's how you build your knowledge. So that's what I've been trying to do. So tonight again, we'll take the idea of prokaryotes and eukaryote. Prokaryote means before the nut, because karyote is a Greek word which means nut or kernel, and it refers to the fact that in a eukaryote there's a nucleus. So that's the nut or the kernel. That's the karyote part of prokaryote. So if you're a bacterium or an archaea, you don't have any organelles, nor do you have a nucleus. All you've got is your DNA lying around in the middle there, and you've got a few other organelles as well, but not much. A eukaryote is a lot more complex. So prokaryotes came first, and they're simple. By the way, I did watch a bit. I haven't watched most of what I've done, but I watched a little bit of one, and I realized that I was talking to the board. I'm sorry. I mean, I'm a teacher from way back. You do not turn your back on an audience and talk to the bloody blackboard. So I'm really sorry about that. That's totally rookie move. Anyway, I had a teacher who did his practice teaching in Glasgow. And according to my grandfather, who was a Scot, Glasgow used to be a really tough town. This guy could write on the board with either hand, and he could look at the audience the whole time that he was writing, and he would never miss a beat. So if Ross in the back row there was, as usual, acting out, he'd nail him right away. Even to this day, when I'm teaching, I tend not to look at the front row. I always automatically look for the Rosses and other bad actors at the back. Never. So if you want to get away with anything in my class, you sit in the front row and I wouldn't even see what you were doing, right? Because I'm so used to people walking in, and I'd say, like, guy would walk in from last year, right? I'd say, "Hi, Craig. Hi, Mr. Horn." He'd sit down. I'd say, "Why are you sitting back there?" He'd say, "That's where I want to sit." I say, "No, but you remember what happened last year? You were there for one day, and you ended up at the front. That's where you're going to end up again." He says, "Oh, well, whatever." And he'd end up again for sure every time. But you get used to it. Anyhow, prokaryotes. So, to sum up, prokaryotes came first. They're simple. There's the DNA, and that DNA is not in a nucleus. The other things that they have is a flagellum. Flagella can either be twirled around to propel you that way, or they could be twirled the other way, in which case they pull you along. So they have, I mean, bacteria are amazing little things. I mean, they are so flexible. The stuff they can do. It's no wonder they're still around. And it's no wonder we haven't been able to beat them. I mean, I didn't know anything about bacteria up until a couple of years ago, but I have a world of respect for the little beggars now, let me tell you. Anyway, they've got a flagellum. That flagellum is powered by the most miraculous little motor. I'll show you one at an appropriate time, but there's a little molecular motor in there that actually turns around. It's a little molecular rotary motor, and these molecules, honest to God, are turning around, propelling that flagellum. It's like a rotary motor made out of molecules on a minuscule microscopic scale. They also have these things on the side, one pilus, several pili. It's pronounced pili because I took 3 years of Latin with Mrs. Smith. And if you pronounced your eyes as an I, you got a steel-edged ruler on the knuckle. In Latin, the I's are pronounced as E's. And there's no soft sounds in Latin, so it's always hard, and it's always with an I. Anyway, that pilus is a Latin word because the Romans used to have these spears. They were short, and they were deliberately made so that when you threw them, whatever it hit would bend the blade on the end so that the spear would become useless. So if you're a Roman there and it's some guy on the, or you're a Roman, and you see a guy over there and you throw your spear, if your spear doesn't hit him, or even if it does, it can't be used again because it's now bent, right? So the idea is bend them deliberately, because otherwise the guys pick them up and throw them back at you. So that's why they were made like that. And that is the pilus, that is a Roman spear. And what the bacterium uses it for is it uses it to conjugate, which is one of the 3 ways in which bacteria can get new genetic information. I will tell you more about that later, and not indefinitely later, but tonight later. All right, so to sum up, prokaryotes, simpler, earlier, no nucleus, no organelles. Eukaryote, it's got a nucleus. That's where the DNA is. It's also got organelles. More on that later, but tonight we're going to learn that 2 of the organelles are the mitochondrion and the ribosomes. And we're going to learn about ribosomes because those are where the proteins are made. So we've all got RNA in us, and the RNA makes proteins, the most important molecule, proteins right there, made of amino acids, most important macromolecule in the universe, because that's what most animals are mostly made of, protein. So the DNA is kept in the nucleus, then it'll be taken out of the nucleus by the RNA, assembled by another kind of RNA over here. No, it's going to be gathered by another kind of RNA, and then it's going to be assembled by yet another kind, and will take a little bit bigger look at how proteins are made by RNA. So that will be part of tonight's topic. So that's about it. I just want, yeah, and the mitochondrion, that's how we get our energy. And we'll do more on this later, but during evolution, now they're not sure where, this is a subject of great controversy, but somewhere down in here in that billion-year period, what happened was one day, a simple prokaryotic organism, an archaea, which is like a bacteria. It's not, but it's small and simple, like a bacteria. An archaea was just going along minding its own business, and along came another prokaryote, and this other prokaryote was aerobic. This other prokaryote could use and produce oxygen. And if you can use and produce oxygen, it's a way better way to get your energy than any other way. It's much more efficient. So this one engulfed the other one that was oxygenic, and the oxygenic bacterium stayed there. Instead of the oxygenic bacterium being digested, it survived. It survived in there, it bred in there, and from then on, a couple of billion years ago, that bacterium has been part of every cell in the body of every eukaryote in the world, including us. So we're sitting here, and each one of our cells, our 37.2 trillion cells, each one of our cells has in it a mitochondrion, and that mitochondrion gives us our energy. And that mitochondrion was originally a free-swimming, free-living bacterium, but they've been in us now for a billion or two, probably a couple of billion years at least. And what they do is, well, we know this. How do we know this? I mean, you weren't there, right? You don't know because you weren't there. Okay. We know because this when this was a bacterium, it had its own DNA, right? Well, it still does. Still got its own DNA. That's why when people talk about DNA, they make sure that they talk about nuclear DNA or mitochondrial DNA. And if you're a woman, you are the one that passes down all the mitochondrial DNA. So that is one way they trace human lineages back, through mitochondrial DNA. That means they're always going through the mom. So that's about it for now. Ribosomes then, we'll be looking at them, and the mitochondria. That process I just described, where one simple thing gets in another simple thing, is called endosymbiosis, meaning living together inside. So I'll just write it on here. Endosymbiosis. Endo, inside, symbiosis, living together. I already told you about bobtail squid and a couple other things that have that do endosymbiosis. There, we've done that. What we were doing last time was looking at what is life. So I'll just go through that quickly, because Denise said I spent too much time on it when I was practicing on her last night. It doesn't sound good, does it? I was practicing on Denise last night. Anyway, wow. Some of these I haven't seen. Denise gets kind of creative. Oh yeah. Oh yeah. That's that. Is that Pyrolobus fumari or
Yes.
Pyrococcus? Oh shoot. Anyway.
That's the Furiosa.
Pyrococcus furiosus?
Yeah.
Yeah. That is the most heat-tolerant organism on the face of the earth. It lives down in hydrothermal vents, down the bottom of the ocean. The water coming out of the vents averages about 400 degrees Celsius, so 4 times boiling. Sometimes it's hotter, sometimes it's a little cooler, but it's a brutal place to live. It's pitch black. It's 2,750 meters down. That means it's got nearly 4,000 pounds per square inch of pressure on it. To give you an idea, a nuclear submarine will crush at 900 meters. These things are living at 2,750. Get this, That thing is most comfortable at exactly 103 degrees Celsius. That's when it's really happy. If you cool it down to 90, it freezes. It can't function anymore. These things are down at the bottom of the ocean, and these things have probably, some of them, been around for nearly ever since the Hadean, that period we're talking about. These are some of the earliest things that there are, and they're prokaryotes. That's a prokaryote. But that thing next to it, you can tell right away. That's looks to me like at the end of a nerve cell, and it's obviously a eukaryote, right? You see all the stuff. You see the nucleus. You see the endoplasmic reticulum. You can see the Golgi body, the whole bit. Anyway, okay, what is life? Okay, it's a quick review. Life has an internal environment. How does it have that? It has it in cells. Why do I keep harping on cells? Because cells are the only thing we've found so far in the universe that house life. You may be thinking, well, what about a bacteria? It doesn't have a cell. That's because bacteria, I mean not bacteria, but viruses, they're not alive. We'll talk about them in just a sec. So, what is life? Life regulates its own internal environment. I like my temperature to be about 37 degrees. I don't feel too good if it gets too much below or too much above. Same with all of us. We have to keep our salt levels, our metabolites, everything in us has to exist within a very narrow range of parameters, or we get sick, or we die. And that's the price we pay for being an advanced form of life. We lose the flexibility that bacteria have, and we pay for it in terms of, well, we have one way of reproducing, right? I think you probably got a pretty good idea by now what it is. With bacteria, you have a whole range of options. You're not limited, right? You'll learn more about this as we go along. So here's your basic divisions of life, and we will be discussing these things. We have bacteria and archaea. They're the prokaryotes. I should get my thing. Where is it? Ah, there it is. Okay. So once again, one last time, hopefully, there's your oh wait. Let me try it this way. I was going to practice, these wouldn't let me. Okay, bacteria and archaea, they're your prokaryotes. Everything else are eukaryotes, and everything else is slime molds, animals, fungi, plants, ciliates, flagellates, whatever these are, and those, and those. You know why I don't want to know what those are? Because we used to call those protists, but these stupid biologists have come along and completely rewritten biology. Now, back in the old days, maybe you guys remember this. You remember King Philip came over for good spaghetti? You don't remember that. Okay. Well, these, you know, you have to. And Claire had this installed just for us, which I thought was really nice. Okay, well, let's take this off.
King Philip came over.
Yeah, for spaghetti.
For good spaghetti.
Good spaghetti.
Yeah, good.
Good.
So, let's say I'm a scientist. I'm going to attend a conference in Russia, and my Russian isn't very good. But let's say I want to have a talk to one of the guys there about an animal, like, I don't know. Let's say I'm a Canadian, so I will go there and I want to talk about beavers, okay? So, I may not know any Russian, and he may not know any Canadian, but if I say that it's a Castor canadensis, you'll know exactly right away that I'm talking about a beaver. If I were to say a rainbow trout, then it would be Salmo gardneri. Except, if he was on his toes, he would say, aha! That was changed a few years ago, and now it is an Oncorhynchus micus. That's why I'm confused. Back in the good old days, things used to have these Latin names, and I used to know the Latin names, all of them, because I decided when I was in grade 5 that I was going to be a biologist. So I remember, I can still to this bloody day tell you the Latin names of everything from moose and elk and deer and beaver and muskrats and mice and all this other stuff, just because I thought I was going to do that. My point is, I don't know what diplomonids or microsporidians are, because they used to be called different things that I used to know back in those days. This has all been totally changed. But anyway, what is life? Life has its own metabolism. Life is enclosed in a cell, and life grows. Now, I think I showed you those salt crystals. Is that correct last time?
Yes.
I showed you those giant salt crystals? They grow, but they're not alive, right? Okay, good. Another thing about life is it has, like, okay, we talk about life interacting with the environment. What we have to remember is life also is the environment, right? Ever since life got started, it has become part of the environment. And that is the reason that I showed you that. Remember I said that if there's no, if you're a planetary body, let's say you're floating around in space, and you've never gotten big enough to get hot, and you've never got big enough to melt, then you are what is called undifferentiated, just a ball of rock. Like, okay, the moons of Mars. Mars has got moons called Deimos and Phobos, named after a couple of Greek guys. And those are so small, they're not even round. They just look like a couple of potatoes floating around in space. So that's what an object looks like when it's not big enough for gravity to make it round. It's got to be a certain size or gravity can't round it out. So when something's 1,000 kilometers, it gets nice and round. Now, I told you last time that if you looked at a globe of the Earth and ran your hand over it, if it were really the Earth, you wouldn't even feel a mountain. You wouldn't even feel anything there. It would be perfectly smooth because the mountains are so small in comparison with the size of the planet. But anyway, life, it adapts. Oh yeah. So if you're undifferentiated, there's going to be 60 elements. If you've been heated and cooled, that goes to 250. If you add water, 420. I'm looking at the board again. 1,500 with tectonics, because things start to get recycled and mixed up, so you get a lot more when you get tectonics. But the big kick is life. 3,500 from life alone. So over half the rocks that are on Earth wouldn't be here if it weren't alive. So growing is part of life, but rocks can grow and they're not alive. They don't undergo Darwinian evolution. They undergo evolution, but not Darwinian evolution. Adaptation, you've got to be able to adapt. And that's where your DNA and your RNA come in. So we'll be looking, when we do DNA, I'll show you a couple of ways that bacteria and other life forms adapt to the fact they've got to keep their DNA flexible so that it can adapt to the environment. Another thing is, if you're alive, you respond to the environment. I use sunflowers and giraffes as a way to illustrate that. Did I not?
Yes.
I did tell you guys about that, right?
Yes.
Okay, good, thanks. Finally, there's reproduction. And if you're a prokaryote, you have one way to do it. You can divide. That's it. You've got binary fission. So if you're a Pyrolobus fumari or a Pyrococcus, sorry, I'm getting the two mixed up. They're both extremely heat resistant. They're both archaea. They both live in the same place, and the Latin names are almost the same. One of them's Pyrolobus fumari, and the other is Pyrococcus furiosus.
Furiosus.
Huh?
Furiosus.
Well, that's
That
what I
one
said,
is-
isn't it?
That one is Furiosus.
Oh, that one's Furiosus. I love the Latin names.
Yeah.
But anyway, same deal as I just showed you down there. The Pyrococcus furiosus is the same as if it's a white-tailed deer, it's Anotocoelius virginianus. Same idea exactly, whether you're a prokaryote or a eukaryote. Let me think. All right, so that's basically it then. If you are alive, you can reproduce, you can respond to your environment, you can adapt genetically, you can grow, you have your own metabolism, you contain it in a cell, and you control your own internal environment. And that's called homeostasis. Okay, what about viruses? Well, that's a tricky one, but viruses are not considered to be alive. And that's why I don't know more, seriously, much about them, because I've been reading about life, and most of, And they don't enter into it. I mean, you can read about life all you want, and there's nothing in there about viruses. When you look at, like I have spent the last 2 years looking at the evolution of life on Earth, viruses are hardly ever mentioned. And yet, they just analyzed the last universal common ancestor, L-U-C-A, Luca, the last universal common ancestor. That is the life form from which all life that we know of sprang. So the last universal common ancestor is that life form from which all life, either fossilized or extant, is thought to have come from. And they just did they, being scientists who specialize in genomics and genetics and paleogenetics, just did a huge study, which has been widely publicized. You'll probably see it if you want to go there. And what it says is that about between 4.33 and 4 billion years ago, there was an organism somewhat like this, living in an extremely hostile environment. It was making its living by chemically altering rocks into a substance called acetate, which is a common macromolecule that's used in a lot of functions, a lot of reactions, a lot of metabolism involves molecules like acetate. So this was a simple organism, but the point is that it was way more complicated than they thought it would be. This is 4.3 billion years ago, when most people didn't even think, I mean, it wasn't very long ago when everybody thought there couldn't be anything alive back then, period. But 4.33 billion years ago, this organism not only had DNA and RNA, but it also had an immune system. Now think about that for a second. It had an immune system. What do we need an immune system for? Denise! Would you like to stay in after school?
To fight other microbes off?
Right. We've got immune systems because things like viruses are attacking us, right? That thing had an immune system because it was being attacked by viruses, and that was 4.33 billion years ago. Now, this is really mind-blowing because this is not the first organism, Luca. All that is the organism from which all life we know came from. There was stuff before that. There had to have been live stuff on Earth millions, I mean many millions of years before that. These are highly evolved creatures. They've got these flagella, and they've got, they've got everything I showed you. They don't have a nucleus, they don't have organelles, but they got a cell wall and a membrane. They got a bunch of DNA, they got a bunch of RNA. They got these things that, these things serve two purposes there, if I remember correctly. One is mobility, but the other is they use them to join together with other creatures of their own kind in order to share resources. That's another thing bacteria do. There's a kind of bacteria called cable bacteria, and they live in sediment where there's no oxygen. What they do is they have got they get together and they form this long kind of like tube. It's called a periplasmic tube, and it's about 3 centimeters long. Now, that doesn't sound like a lot, right? 3 centimeters? But if you're a bacterium, that's a long way, because your average bacterium's about 1 to 2 millionths of a meter long. 1 or 2 millionths. So 3 centimeters is a long way if you're a bacterium. So what these little devils do, at the top of let's say there's a layer of sediment. And at the top of the layer of sediment, there's oxygen. Everybody wants oxygen, right? Oxygen is the world's best electron acceptor. So if you're going to have an electric current, you need two things. You need an electron donor. You got to get an electron someplace. You got to have an electron receiver or acceptor. And between the two, you get an electric current, right? You get electrons flowing from your donor, which is usually hydrogen, to something like oxygen, which is usually your acceptor. So you now get an electric current going. And what these things do, they get together and they take the oxygen. So the oxygen is on the top up here, and down below there's no oxygen because oxygen does not penetrate sediments well. So as soon as you get down to sediment, no oxygen. You've been to the beach, okay? You've been to the beach, low tide, dig it in the sand, you overturn this muck, okay? This muck, and the muck is kind of black, and it smells kind of like rotten eggs. That's hydrogen sulfide. That is called the anoxic layer of the mud. So on the surface of the mud, where you're standing, there's oxygen. A little way down, where it's black, there isn't. What there is down there is hydrogen sulfide. So these little organisms, bacteria, what they want to do is they want to use the oxygen as the electron acceptor, but they need something to be a good electron donor. Well, the best electron donor they can find locally is buried down a few layers in the mud. So they use that as the electron donor, and the oxygen up top is the electron receiver. And the bacteria form a bacterial, it's like a human bucket brigade or something. You know, get the idea? The bacteria all get together and they form this tube between the oxygen and the hydrogen sulfide. And it's only 3 centimeters, but for them it's like thousands of miles. Isn't that something? And that's how they beat the problem. I think that's just, you can see why the little devils are so hard to kill. They can do just about anything. I'll tell you, wait till I tell you about some others. There's others that live off electricity, purely, nothing else. They just live off electricity. Remember I mentioned Pavilion Lake? There are bacteria they have now discovered. I was just reading this last night. SFU was doing a study out there at the same time as NASA, and SFU has now found magnetotactic bacteria in Pavilion Lake. And magnetotactic bacteria, which they think have been around ever since this time we're talking about, they're probably one of the first life forms. Those magnetotactic bacteria have in them little tiny bits of magnetite. Now, if you're driving from here to Kamloops or someplace, if you're like me, you're always looking at the road cuts because you've always got your eye out for minerals, right? And if you see these kind of blacky, gray colored minerals, which are really common, they've often got some brown or red mixed in. Those are iron oxides. And the most common iron oxide is this one. That's magnetite. So that's what magnets stick to. That is the ore in the world that magnets stick to. And that is made out of ferric oxide. So, oh yeah, that's what these magnetotactic bacteria have got inside them. They've got, that's called magnetite. And again, it's an oxide of iron. It happened when iron met, wait, I've got this thing in my hand. I'm pointing it futilely. Let me see if it, yeah, so there's your iron. That gets oxygen meets iron, and you get magnetite, and that's what you see in the road cuts. It's that really common blacky, grayish colored rock. That's magnetite. And that is what those magnetotactic bacteria have got inside them. But they're so small. I mean, a bacteria is 2 millionths of a meter, right? Well, these are small enough to fit in the bacteria. There, look at what Denise did. She got us a picture of those stromatolites in Pavilion Lake. It's one of the few lakes in the entire world where these things are found. So that's why it's such a popular area for research. We're on the map, ladies and gentlemen, because of the camels and because of Pavilion Lake. Okay, so viruses are not alive. Why? They're just a set of genes and a protein coat. They're not alive because they don't live in cells, they don't metabolize. When viruses are created, they're created fully formed. Like, they don't grow. They're as big when they're formed as they ever are. That just seems weird to me. The other thing that seems weird is that I can be killed by something that isn't even alive. But I guess it's not that weird. Anyway, so viruses aren't alive, and that's the story on them. So now we know what life is. Now we're going to look at what early Earth was like. Oh, yeah, okay, I gotta tell you this first. I'm actually digressing here, but not right at the moment. First of all, I'm gonna tell you something. According to that should be 87. Is that right, Diane?
Yes.
Okay, that's a virus.
You want the zircon?
Yeah, let's have the zircon, please. Okay. More about the zircon in a minute when we do Life on Earth, but I just wanted to tell you about Galvani, Volta, and Frankenstein, okay? Because they do directly relate to Life on Earth. Right? I'm being a bit defensive here about my digression, I think, but Denise has really tried to talk to me about it. Okay, so, all right, there he is. Back in the 1780s and 1790s, this Italian, not that his name Luigi Galvani, I mean, it could be Scottish, I guess. But anyway, Luigi Galvani in Italy was experimenting with electricity and animals. Specifically, one day, there was a thunderstorm, electrical storm, and Galvani took a piece of metal and he applied it to the leg of a dead frog, and the dead frog moved. So he started to realize there was a connection between electricity and muscular contraction. So he started doing experiments, and he was found that, yes, he could get dead frogs to move all over the place by applying electricity to them. And so he came up with the idea of animal electricity. He thought it was a special kind of electricity that animals had that enabled them to move. So people are trying to figure out what life is all about, right? Okay, so Galvani does these experiments with frogs, comes up with the idea of animal electricity. Volta, after whom the volt is named, obviously galvanism is named after Galvani, right, and the term galvanized. Anyway, Volta, after whom the volt is named, didn't agree. Okay, fast forward now a little bit to 1816. In 1816, we're in Switzerland now, ski country, Mary Wollstonecraft, brilliant woman, her father was a world famous philosopher. Mary Wollstonecraft, this isn't her boyfriend, that's not Percy, was with her soon-to-be husband, Percy, the poet. And they were holidaying in Switzerland, and they met up with the Byrons, Lord Byron and his latest flame, who I think at the time was his girlfriend and half-sister Claire Claremont. Anyway, the Byron couple and the Shelley couple got together with some other people, and they had a story writing contest to pass the time because the skiing wasn't very good. Mary Shelley wrote a story called Frankenstein, and everybody agreed that it was the best story that it was written. It won the contest. And Mary Shelley had been to Frankenstein Castle before that, so the name Frankenstein actually has a real, honest-to-God, historic provenance. So she wrote this story, Frankenstein. It was a short story, it's a pretty good story. But then she took her husband's advice, which was terrible, and made it into a novel, which I couldn't stand. But anyway, it made a famous novel and a famous movie. And yeah, I used to teach it for AP, but I finally quit. I just couldn't get behind it. It was too, I don't know, just too, I don't know, never mind. The point is that what Mary Shelley was doing in that book was employing the idea put out by Galvani, that there was this thing called animal electricity. And the idea was that well, in the book, Victor Frankenstein, with the best of intentions, goes out and gets all these body parts, fresh body parts, and puts them together. Then he puts electricity through it. And of course, he does it at night, right? He doesn't do it in the middle of the day. Does it at the scariest time, right in the middle of the night. Of course, the creature's yellow eye opens, right? There's a famous scene. It's kind of creepy. Well, she's employing the idea of animal electricity of that, and that is not a right idea. The idea of electricity is good because it's electricity that's enabling us to sit here and be alive right now. If we didn't have electrons flowing through our membranes operating proton pumps that were firing proteins across a membrane and causing a gradient and a differential, and then if it didn't allow those to go back in through a little ATP engine, we wouldn't be sitting here, we'd be dead. If you stop that process for 1 minute, have you seen the thing on TV where somebody takes cyanide and they foam at the mouth? You've all seen that, right? Well, that's what we'd all look like in about 1 second if that process quit. That's why cyanide works like that. It stops that little machine that's pumping protons from one side of the cell to the other. Cyanide stops that, one of those little machines from working, and we die instantly. Yeah? So that's the idea, animal electricity and all that stuff. Now, to get down to the real science, fast forward ahead now to 1952, we're going to look at Miller and Urey. Okay, young graduate student named Stanley Miller attended a lecture by the world famous laureate Harold Urey, and was inspired to go up to Urey after and say, look, I think we should work together and try an experiment here. Let's reproduce as best we can what the conditions were like